Hello everyone, welcome to Science Nerd. Today we're going to talk about how to determine the molecular weight of proteins. So the first question is, what are proteins made up of? They're made up of amino acids. So various amino acids, when they're bonded together, they form what we call as proteins. Now, by default, the weight of amino acid, we take it into ratio with carbon 12, so it is 128 Dalton. But whenever you see the MCQs or whenever you study, it is not 128 when we consider it in the peptide chain of a polymer of protein. So why is it the reason it is not 128? Because when the bonds are formed, there is a thing called as water molecule which is removed. Minus H2O means H2O is removed. So 128 for the amino acid. And what is the molecular mass of H2O? It is 2 molecules of H. H has 1 and oxygen. Oxygen is 16. So 2 plus 16 is 18. So 128 for amino acid minus 18 gives us 110 Dalton. Now you may have come across another term called as amino acid residue. What is the difference between an amino acid and an amino acid residue? When there is a water molecule is removed, that is amino acid minus water molecule gives us amino acid residue. So amino acid residue is simply an amino acid without a water molecule. So I think this is clear. This is the basic concept of how to calculate the molecular weight of proteins. Now we will move on with some kind of questions so that we understand the concept really well. So these are the questions which are found on Pathfinder as well, and there will be many more questions you can find and then you can try. So we'll do these two questions for an example point of view. A protein X of length 1000 amino acid. There's a protein X, length is 1000 amino acid. It is fused with GFP. GFP is green fluorescent protein. The length of GFP is 27 kilodalton. Find the molecular weight of fusion protein. This is the question. So it says there's a protein called X and then there's a protein called GFP. We fuse these two together. This is what we get. A fusion protein, that is these two fused together. So it says that the length of protein X is 1000 amino acids. So it is, what is 1000 amino acid? X is 1000 amino acid. So as we studied earlier that the molecular weight of amino acid is 110. Whenever it is not provided in the question, always assume this as the molecular weight of the amino acid. So in this, so we'll write 1000 because of this. There are 1000 into 110. Otherwise, we could have taken it as 128 into 1000 minus then 27 if it was linear. And if circular, we would have taken 28 H2O removed now. So this is the way. But since it is not given linear circular, we will consider it as 110. So what is the answer we get? We get 11000. Yes. So this comes to be the molecular weight of X. Clear? Now for GFP, it is already given 27 kilodalton. To convert 27 kilodalton into dalton, we can write as 27,000 dalton. I hope this is clear. So we'll take another marker. X is this, and this is for GFP. So we can add these because this plus this is the fusion protein. In this we will add 27,000 plus, how much do we get? We get, so this is your answer, and your answer will always be in dalton if you write like this, or if in the option that is given kilodalton, or if you're asked to write in kilodalton, then it is 137 kilodalton. So this is your first question. So for the next question, we have to learn a concept again. So it is a very simple concept. I just mentioned it here as well, and it is about linear and circular. So it says the molecular mass of arginine. Arginine is written as R. So if you do not know the one letter codon, you always need to remember these, because sometimes they may not give the 3 letter codon. They may only ask from the 1 letter codon. Sometimes they may even write it as complete. So just in case you do not know how the question will ask you, you need to know the 1 letter codon, and for the 1 letter codon you can watch my first video on mnemonics of amino acid. In that I've talked about the essential, the non-essential, the semi-essential, the hydrophobic, hydrophilic, polar, non-polar, and basic and acidic amino acids. So you can check that out. Moving on, it says arginine has molecular weight of 174 dalton. And now it says find circular polymer of 38 arginine residues. Okay. So what is linear? I'll take 1, 2, 3, 4, 5 amino acids making a protein. Okay, and in circular case, amino acids can join like this as well to form protein. So then this is a linear arrangement, and this is a case of circular arrangement. In this case, if we say there is 1 bond, 2 bond, 3 bond, 4 bond. 4 bonds but 5 amino acids, and in this case 1, 2, 3, 4, 5. 5 bonds and 5 amino acids and only. So for the case of linear, the bonds will be n minus 1, and for circular it will be n. So the number of bonds, the times the H2O will be removed. So this is the concept. So it says there are 38 polymers of arginine. So we'll do it as 174 into 38 minus 18. Why? Water is 18. Okay, and R is this 18 into 38. Here we get it as 6612, and here it is 684. You get it as 5928 dalton. So this is your answer. So this is a very simple concept, but it is very important for the point of view of competitive exams, CSIR-NET, GAT, DBT, ICMR, for your undergraduate courses, your college exam, your PG exams. So it is very important. I hope you liked the video. Thank you. Do subscribe to my channel.